Hi, welcome in Module 2, Lecture 2. In Lecture 2, we're going to look at new sequencing methods other than Sanger sequencing. So what are the new sequencing methods? They are the emergency sequencing methods listed here, and we're going to discuss each of them. So we're going to discuss sequencing by hybridization, the mass spectrometry techniques, nanopore sequencing, direct visualization of DNA, the Polanyi sequencing, and pyrosequencing. So let's start one by one. Sequencing with hybridization is practically using oligonucleotides, which is on a chip, and those oligonucleotides has certain sequences which can hybridize the sequence you want to DNA sequence you want to sequence. So you have this little array of chips of short sequences, and that will be hybridized to DNA sequence. All right, let me show you the. So here's your chip. You have oligonucleotides, short DNA sequences on the chip. It can actually represent a variation of a chromosomal region, okay? And you will see which one your DNA fragment will hybridize to the sequence. Now, this technique is not good for new sequences because we have to know the sequence. But this technique is now very popular to finding variation in the genome, and because you could have this huge plate, you could have a lot of different possible sequences. And so, for example, a good application for that one, if you want to find a variation in, let's say, a gene, and you have this, let's say, 1,000 different genetic fragments from different people, you can practically screen them in one experiment. So again, that's called the sequencing by hybridization. And when we're looking at the DNA chips later, this course, we're going to talk about the SNP arrays. And the SNP arrays are now extremely popular and extremely well studied, and practically they're using this technique. So again, sequencing by hybridization is not finding new sequences, but verifying existing sequences and or finding variations. I'm sorry about my. The next one is, which is getting also very popular, is the mass spectrometry DNA sequencing. Mass spectrometry is, of course, the machine from the 1970s, and we used it for a lot of different purposes, but recently we discovered that we can sequence with mass spectrometry, and even we can have very high resolutions of the sequencing fragments, very rapid. It can take microseconds. It can sequence up to 50 base pair. Now, you can say 50 base pair is not long, yeah, but if it takes only a couple microseconds, you could go very fast on this chromosome. We don't have those compression, those GC compression I mentioned before, as we have the gel-based system. And practically those fragments will provide a spectra, almost like a fingerprint, the mass spectra, with very good resolution, as I mentioned. So if you're looking for mutation or single nucleotide polymorphism, like the SNPs, extremely valuable method. And actually it's very cost efficient. So this is how it looks like. So practically, here is what you see after as a result. Okay, we're going to talk about mass spectrometry in a later lecture. I'm going to explain what is MZ ratio, but let's just look at right now how it comes out from the machine, and practically after you could assign nucleotide for a certain position. Again, a very upcoming technology and very promising technology. The other promising technology called nanopore sequencing. So what you do in nanopore sequencing, you're gonna take your DNA and you're gonna pull it through a nanopore protein channel like alpha hemolysin. So what happens when you pull your DNA through this channel, the nucleotides will block ions from moving through. So you can measure the differences in the currency. And the techniques is still needs improvement, but the last time I heard is now they have some alpha or beta testers For example, my husband works for PerkinElmer, and they got a nanopore machine, and they right now beta testing whether it is accurate. Very fast sequencing. It can do 1,000 bases per second. So again, another very promising technology. And this is just a picture of it. So you see that practically the channel, you get it your DNA helix through, and that's gonna change the current, and you can measure the current, and from that one you can practically get the sequence. Again, very upcoming technology. Hopefully we can see it in the near future. The other upcoming technology is called the atomic force microscopy technique. What you can do with atomic force microscope, you can actually see DNA. And this is an atomic force microscope. Atomic force microscope works like a blind man. It's tapping the samples, but the good news is it has extremely high resolution. Right now, resolution is very close that we were able to see the DNA bases, the nucleotides. So again, the technique still needs some modification, but again, if we were able to visualize the DNA, that with a special camera we were able to do very fast sequencing. So this is the techniques which still needs some improvement, but one of the techniques which are already in use called the clonal amplification on DNA fragments on a solid surface. Okay, and this usually combined with the PCR method, and from this clonal amplification you can get sequences parallel. I'm going to explain a little bit, just 1 second. The other one is the pyrosequencing, which helps to sequence these clonal fragments. Now, so how this whole thing works? Those clonal fragments actually they call the Polony. So Polony DNA sequences. So what they try to do here, instead of reading one fragment at a time, they're gonna read millions of fragments at a time, parallel, at the same time. And doing that, you could get tremendous amount of data parallel in every experiment. Makes this technique very inexpensive and accurate. So practically, remember the shotgun libraries? You could make the shotgun libraries, but you made the library on solid surface, like beads. There are special beads which you can practically attach your DNA. And after using a special PCR method, you can amplify it on the beads. So every bead will have millions of copies of the same sequence. Okay. If you some technique actually using as a surface some microscope slides. So whether you put on beads or you put in microscope slides, same idea. Having amplified DNA, millions of copies of usually short fragments, but those copies are read simultaneously. So every second you could have millions of data. Of course, you need a computer to process this, but now you can sequence very fast. Okay, so here's how it looks. So you have your fragment. Okay. You put your fragment on a solid surface. That could be a slide, that could be a bead. Okay. After, use a special PCR method to amplify, make millions of copies of each fragment. Okay. And after is a special procedure when you in situ, so in place, you're gonna sequence them. So every fragment will sequence the same time. Remember, you have millions of these fragments. Okay. I explained to you how to sequence it. So every steps you get data from every single fragment. All right. The famous company who is right now the leading company in this Polony sequencing is called Illumina. So what they do, this is just a summary from their website. What they do, they have here's the DNA, they fragment the DNA, they put two adapters on this fragmented DNA. After they put this fragmented DNA on a surface, as you see, on the surface you already have the adapters attached. So the DNA gonna bind to those adapters, okay, all right, because they are complementary, right? And they're gonna have they're gonna put as many fragments that they have a good representation of each fragment. After the special techniques that these fragments actually gonna bind. See, remember it has two adapters, so this fragment gonna bind to the adapter close by, making sort of this bridge structure, and the bridge structure will be amplified by PCR technology. So doing this, let's go back a little bit. So this is the adapter. So the adapters which contains the fragment. This is the DNA fragment. As you see, we attach the DNA fragments for the surface, and after you do this, the DNA fragment will bind to the close by adapter because they're complementary in sequence, and we can do the bridge amplification. Okay. After, you're gonna have this Polonese, yeah, so million of copies, okay, with the same sequence, and now you're gonna send the labeled nucleotide, right, primers of course, and polymerase, and you start to sequence it. And what you could do, every time something binds to it, it's gonna send out a signal. It is fluorescent dye, so either a red signal, a blue signal, a green, or a yellow signal. And the machine actually watching this plate and records which nucleotide gets in. So that's called a sequencing by synthesis. Okay. So for example, here's just a second of a cycle. As you see, we see in couple Polonese, and this Polonese has an A, looks like this Polonese has a C, this Polonese has a T, and this Polonese has a G put in it. So now the laser can read this and can send the information to the computer, and the computer records the information. And this is was just a second. And the next time, the next second, when is a new nucleotide gonna come in, obviously records it also and builds. This is cycle 1, cycle 2, cycle 3, cycle 4, cycle 5. So now, for example, for this Polony, we have a sequence GCTGA. Okay, all right. So this is how the Polony sequences works, and it's amazing how accurate it is. Again, my husband working is for a company who using Illumina technology, and they can sequence now human genomes within a day or two, and the accuracy right now they have, I think, is minimum 50-fold, but depends on, you can go up as much as 100-fold, which gonna give you 99.9999% accuracy. Okay, so this is now the current technology, and on your assignment 1, I want you to further researching these technologies. There are a couple of variation, what kind of surface they use, what kind of technology to use for sequencing, what is the reads out. So assignment 1, you're gonna research it, and you're gonna make a summary of these new technologies, because this is the upcoming technology which is used right now. One of the sequencing methods which was developed back in 2000 in Brantford, Connecticut, believe it or not, right here around the corner, in something called 454, a company called 454, that's called the pyro sequencing. And the person who established 454, his name is Dr. Radberg. And Dr. Radberg has lots of different discoveries that was the first major discovery that he was able to using a completely novel technology which was able to combine this Polony sequencing technology together. So again, sequencing by DNA synthesis, as I mentioned, again can be parallel. So you could see those million of polonies in the same time and record the sequence data. Of course, very easily automated. Eliminates the need for labeled primers or nucleotides. Yeah, no need for gel electrophoresis. Now, let me explain how it works. The major enzymes, which is works with this technology called luciferase. Luciferase is an enzyme which emits light in the presence of ATP. Okay, for example, American fruit firefly or the jack-o'-lantern mushroom can produce luciferase, so which means they can glow in the dark. Again, that was developed right now called the Life Science or Roche 454. They all using these technologies. But again, developed in 454 with Dr. Rodbeck. So how can we make the light? So pyrophosphate is also known PPI, two phosphate groups stuck together practically. During replication, when you add DNTPs, the food for replication, when the DNTP is built in, it releases the pyrophosphate. So the pyrophosphate allows adenosine phosphosulfate APS to be converted to ATP. And if it converted, ATP actually can activate luciferase and emit light. So how you use this? So you first add, let's say, DATP. Okay, if the light is emitted, your sequence starts with A. If not, you have to get rid of the DATP and either you wash it out or you will see you can use an enzyme. After you add DGTP, if the light is emitted, the next base is G. And so on, and repeat until you have the whole sequence. So, again, the basic idea, visible light is generated and is proportional to the number of incorporated nucleotides. For example, 1 picomole DNA can make up 10 to 9 photons. Okay, so you can measure this. What kind of enzyme you need? Of course, you need DNA polymerase from E. coli. You're gonna need, of course, you're gonna need ATP sulfurase. Okay, and you're gonna need luciferase. All right. So here is the summary. So when the nucleotide builds in, releases pyrophosphate. Pyrophosphate plus APS in the presence of sulfurase makes ATP. And ATP plus luciferin plus oxygen and plus luciferase gonna make light, and you can measure the light. Okay, so that was the first method. Okay, it has the 3 enzymes just I mentioned, and you have to wash washing step to remove the nucleotides after each addition. Okay, so this is what you see in the computer. The second method added a 4th enzyme called apirase. Okay, apirase actually degraded the nucleotides, so you don't need to add wash. You can eliminate the washing step. All right. So of course you need a camera. Remember, it's reading every single polony, like we mentioned in the Illumina polony sequencing, every single polony, and reads after each step whether it's an A, whether it's a C, G, or T. And this is what you see. So for example, here we added T, so it made the T. Okay. If it's a higher peak, for example, here you see the highest peak for T because it was 2 T's. Here was even higher for 3 because it was 3 C's, and so on and so on. All right. So this for the pyrosequencing. I uploaded a very nice video how the whole thing works. Please watch the video also. And I just want to mention this very interesting that the X Prize Foundation in 2006 called for a full genome sequencing technology for the first team who can sequence 100 human genome within 10 days or less with accuracy of no more than one error in every 100,000 bases. So it was 2006, and believe it or not, that was cancelled because it was just the whole technologies developed so fast. So it was a lot of people were able to do this, and they didn't really have any challenge to have this goal. And because that, look at the cost per genome, how much it dropped from 2001. So cost per genome was in $100 million range. Look at that, $100 million. It's now about six to eight. That's a 2013 data. I think now it's 2015, probably more like 5 to 6 or even less. Depends if you sequence just one genome, but if you can sequence multiple genomes, the cost is even less. We have done a sequencing project for one of the clinical studies we did, and we were able to. We had to give them I think 3 genome, and I think the cost per genome was about $4,000 at this point, and it was 2 years ago. So the cost, the sequencing cost is less and less, and of course the goal is to push it under $1,000. So you can sequence your genome practically the price of an MRI test, which is right now $800. All right, so hopefully with these new technologies, we were able to get more and more sequences and able to use these sequences for understanding better the human genome. Thank you very much.